Hello everyone. Welcome back to Dr. Liu's VCE Biological channel. Today, let's learn together about the 2020 VCE Biology. We will focus on questions about the relationship between nucleic acids and proteins. My explanations may need to be revised. If any teachers, parents, or students find errors, please feel free to provide your guidance. Although the same codons code for the same amino acids in almost all living organisms, the same protein might have different amino acid sequences in different all organisms. Degenerate means multiple codons can code for the same amino acid. MET has one genetic codon, so the genetic code is not degenerate concerning MET. The codon GGU is for GLY, not TRP, so it cannot add TRP to a polypeptide chain. The DNA template is an antisense strand, and the sequence in the antisense strand is complementary to the genetic code in mRNA. The code of Lu is CUU, so its sequence on the DNA template should be GAA. Please note the direction of the sequence. 5' in mRNA corresponds to the 3' in the DNA template. This question is about the role of the operator in the LAC operon. This operator is a sequence in DNA and is not translated into any protein. If the repressor protein binds to this operator, the downstream genes are not expressed. Although the regulatory genes are not translated into structure genes, they also are translated into transcription factors. So, option A is incorrect. Label the 3 subunits on the diagram of the RNA nucleotide. All 3 parts must be labeled: phosphate, base, and ribose. Describe the role of RNA in a cell. tRNA brings an amino acid to the ribosome, or complementary anticodon binds with the codon of mRNA. mRNA carries genetic information from the nucleus to the ribosomes, or complementary copies of the DNA. Outline 2 events that occur during RNA processing and the importance of each event in gene expression. Introns are removed, which are non-coding sequences. Exons are spliced together, they provide the code for protein synthesis. Poly-A tail is added, which facilitates the export of final mRNA from the nucleus, or protects mRNA from degradation. Methyl or guanine cap added, which protects mRNA from degradation, or helps attach mRNA to the ribosome. Exons may be rearranged or changed during splicing before mRNA leaves the nucleus, increasing the variety of proteins and thus reactions that can occur in cells. The diagram below is a simplified example of a signal transduction pathway. Structure X is located on the cellular membrane and binds to a signaling molecule. So, it is a receptor. Structure Y wraps the nucleic, so it is a nuclear membrane. Step 1 is reception. In Step 1, a signaling molecule binds to a receptor. Step 2 is transduction. In Step 2, a series of chemical reactions during which the signal is transferred. Signaling molecules generate a series of messengers that result in activation or deactivation of genes, or the signal is amplified. Step 3 is the response. In Step 3, a protein is produced, or a transcript is produced or stopped, which causes a change in cell function. The molecule is hydrophilic as it does not pass through the plasma membrane, and it attaches to a receptor on the membrane. In the relationship between nucleic acids and proteins, 2 multiple-choice questions are worth 2 marks, and 2 short-answer questions are worth 15 marks. The overall score for this section is 17 marks. With a total possible score of 120 points, the percentage for this section is 14.2% of the total score. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.